So now open for questions from the audience. As you're composing your question and coming to the microphone, let me pose a question to both Ken and David. So there have been great advances in both multiple myeloma and non-small cell lung cancer with response rates using the respective therapies approaching 100%. So what does that mean for the development of new agents for these diseases using early indicators of activity if your frontline therapies are so effective? So Ken, why don't you start, and then David, turn it over to you.
This is one of the happiest problems we've ever had. There are many answers, Mace, but one is that we need to come together as a community, and we have actually under the auspices of the NIH Foundation, and have just completed a project, a white paper on minimal residual disease. So we've actually come together. That means investigators, both basic and clinical, companies, biotechs. The FDA actually wrote sections on how MRD could be useful for regulatory purposes, and then there were clinicians that wrote how it could be useful for clinical practice of medicine in myeloma. But just to cut to the chase, if you came now with a triplet therapy and you treated a new patient with multiple myeloma, there's progression-free survival that's probably 7 to 10 years. And if you're going to try to improve that by 50%, even I will be old at that point. Since I'm already old. But anyway, we can't do that. Patients can't wait for that. We can't afford those trials, et cetera. So we really need something, Mace, the MRD metric. We have some already here and some coming that will be able to tell us, say, at 18 months, what will be the outcome at 10 years. And I think the FDA, to their credit, we've put together the world's experience now. We're going to bolster it with some company de-identified data. But I do think within a not too long, we'll have a robust database that will allow us to try to do that. But you're absolutely right. If we don't do that, it will be nearly impossible to develop a novel agent for newly diagnosed patients.
So raising the bar for the endpoint. David, your perspective?
Sure. I'm glad I got to go second, so I wouldn't be corrected. I agree with Ken. I think, it's very interesting. I look at the paradigm of Hodgkin's disease and testicular lymphoma as what we're basically saying, which is we're going to reach a point with whatever set of agents reaches some maximal benefit. And then the development question starts to shift. It's what can we take away? What regimens can we do that are actually safer? And maybe even look at scheduling when you look at the totality of what patients are getting. So I think the framework of the kinds of questions, and certainly the endpoints to assess these, is what has to evolve. But I also share Ken's enthusiasm. It should not be a signal to stop development in these diseases until we've achieved cures. I think we still have a mission to further development.
Mark Retain, Chicago. Just listening to this, I'm going to put in a plea for more quantitative oncology, quantitative clinical pharmacology. This is clearly an area of interest both in industry as well as the agency. And I think with good models, one can make drug development decisions early. I'm not suggesting that we're going to use a whole bunch of modeling to find surrogate endpoints that are going to be sufficient for approval. We might find endpoints that might be sufficient for accelerated approval. But I think just as an example, there's been work done looking at modeling outcomes, early outcomes in randomized clinical trials. For example, time to tumor growth, if you can show a difference in arms and time to tumor growth. That work is by Renee Bruno and colleagues. Renee's now back with Genentech. That's one example. I would think myeloma is absolutely prime for this. I mean, it's a disease with more quantitative data than any disease I know. We struggle to get tumor measurements, and when we get them, we decide that they're not very good, and there's a push towards volumetric data and completely reanalyzing imaging, but we have all of these paraprotein data, and it would seem to me that there's a great opportunity to mine the data that's been accumulated over the decades in myeloma to really find good ways to develop drugs more efficiently in myeloma.
Thank you. And any responses or more of a statement? Okay, Rich?
So, Rich Shilsky, ASCO. If you look at the title of this session, which is Flexible Drug Development and Decision-Making, Accommodating New Insights, I wanted to ask the panel members to reflect on that a little bit, and maybe also to put it in, to bring in the context that we heard a lot about this morning, which was this notion that we should have pre-specified adaptive designs, which to me sounds like a little bit of an oxymoron, because part of the issue of being able to adapt your trial design might be in response to information that you can't foresee, can't anticipate, you become aware of during the course of the trial. And so it's a little hard to be able to say that under every circumstance you can have a pre-specified adaptation plan. So all of you have been involved in lots of product development, lots of clinical trials, and certainly not everything went from A to B exactly as intended. So I'm wondering if you can give us any examples of new information, unexpected information that came to your attention during the course of a clinical development plan that resulted in a substantial or significant modification of the plan, and how you accommodated that.
Just a couple comments, Rich. I think your question's very provocative. The rationale, at least from a methods point of view, for pre-specifying the adaptations is to keep the errors under control. And I don't think it's clinical trial designers and executors that are solely the ones concerned with those error rates. I think regulators eventually are as well. But of course there are examples where things didn't go as planned. And I've seen many of those. One that came to mind when you mentioned it was the need to terminate recruitment of a subset of individuals in a trial that was unplanned for. That's relatively easy because it didn't perturb the error properties of the rest of the study. On the other hand, many adaptations, unplanned inflations of sample size and things like that run the risk of letting the investigators lose control over the error properties. So from that point of view, I would urge some caution with unplanned adaptations, just as a general principle.
I would comment. And I guess it depends on what context this study is. To me, a randomized phase 2 study is to limit the risk to phase 3. And obviously it's nice when statistics hold up, but if there's a strong hypothesis, scientific rationale, and there's a strong trend, even if the statistics don't hold up, and this is not for registration from a phase 2 study, but just to limit the risk going into phase 3, I think that's acceptable. And in the context of the CDK program, that trial underwent quite a bit of evolution from a single all-comers, 150 patient phase 2 to then being broken into 2 different arms based on biomarkers, and then closing that second arm because we saw the biomarkers weren't important, then folding that back into a single arm analysis plan. And as it turned out, the degree of benefit was great and it met all its statistical endpoints. But the main point of that study was to give us confidence to do the phase 3. And so I think we were very adaptive to new info as that came in.
Can comment. We've done this a number of times. The form of adaptation is usually we have maybe a randomized study that's ongoing. Prior to that, there might have been some single-arm study. And in the context of that single-arm study with more mature follow-up, we make observations that then impact changes to the design of the ongoing randomized study. So it's data external to the randomized study is used to guide a modification. So this could be a change in endpoint or resizing of a study. But that's we've done this a number of times, that form of adaptation. I'm not a big fan of adaptive designs particular for phase one. Part of this has to do with just there's so much we don't know, especially about new molecular entities. And in any of the adaptive studies, there's usually a feedback loop. And you can design, you can build what those rules are. A lot of the times, if it's an efficacy readout, you're having to wait a certain amount of time to get that information and feed that back into the system. And my experience has been that adds an enormous amount of time onto the learning that goes on, unless you choose some surrogate endpoint, maybe it's through medical imaging, that you can get a much quicker answer. Otherwise, I don't see that it adds a lot of utility, and there's an opportunity to have false negatives, to be stopping something too early.
I would say is this, it's not truly adapting a given trial, but it is altering the course of drug development. In some of the early studies of the proteasome inhibitor, there was initial enthusiasm for combining a proteasome inhibitor with a JNK junk inhibitor. And in fact, the preclinical studies and actually the correlative science showed that if you did that, you would block the death signal triggered by the proteasome inhibitor. So I think it's really valuable that a negative result actually saves a clinical trial, et cetera. Conversely, although many pathways are impacted, AKT signaling is activated. So in the place of those junk inhibitor trials came AKT inhibitor trials with proteasome inhibitor, both in myeloma, prostate cancer, and others. But I do think what I'm getting at is sort of the correlative science that goes along with one clinical trial can really inform going forward, not only in one disease, but more broadly.
Keith? Yeah, a couple of comments for a few of the panelists to potentially respond to. So one is that I think the issue about endpoints and evolving endpoints and being flexible needs to have some specificity to it, and I think it came up a little bit. In Ken's comments, MRD that's a myeloma-specific concept to try to lock down that as a early metric of success. In immuno-oncology, I would argue, I was expecting David to bring this up, that the plateau of the curve is kind of the value proposition that many in the field point to say, well, that's what's so compelling about that data set that draws me to PD-1 antibody-based therapy. And one could argue if the dust settles in a given malignancy, you have most of the patients who are benefiting declared by 12 months. Then 12-month PFS landmark comparison between arms, even a small study, becomes a specific strategy. But it's different based on different contexts. I've made the argument for the past few years in BRAF mutant melanoma, it's complete responders just by silly RECIST criteria that do massively better than partial responders. And we have 18% of patients having complete response to BRAF-MEK combination therapy. So for us, I've made the case that if we can just focus on complete response rate in early studies, now that we're looking at BRAF-MEK-based triplets, that would get us a long way. But not suggesting that's going to work for IO combinations or in multiple myeloma. So I think specificity by mechanism as well as disease area are variables that are going to expand and get more complicated, and I don't think we should be trying to create a single platform. And I guess the final comment on specificity is more leveraging Steve's talk. With regard to combinations, it gets pretty ridiculous pretty quickly to try to explore doublets and triplets as though each drug is pulling its own weight the same way. And I have argued that we need to be biased in our dose exploration. So if you were to tell me in a MAP kinase pathway-driven disease, like melanoma, that in a MEK PI3K combination strategy, I need to equally weight a PI3 kinase inhibitor's contribution when preclinically and clinically those drugs won't do anything on their own, I'd say, no. I need to know what dose of that other agent I can give with something that's in the business end of the range for MAP kinase pathway inhibition. That type of strategy per biologically defined subgroup of disease, I think, will be a much more efficient strategy than exploring full surfaces. But it didn't happen with a broad class of MEK PI3K combination trials, is how I would summarize that frustrating experiment.
So I'll just respond quickly to your last remark. I certainly would have no objection to being biased. Just don't be wrong. The other point I would make, you know, we have an embarrassingly large number of potential outcomes. If you spend any fraction of your time outside of oncology, which I do, there are many diseases where they're struggling for their first decent outcome. We can see it under the microscope, we can cut it out, we can measure it, we can watch it grow, we can cultivate it in a jar, we can dissect it, we can map it genomically. I mean, we can do so many things to cancer that other diseases don't have available to them. And you don't need to have a perfect efficacy outcome to accelerate development. You can have a pretty lousy surrogate, but it can still accelerate development. So we should be kind of overjoyed that we have so many things to dig into.
I wanted to go back a little bit to the comment about, you know, having pre-specified endpoints and pre-specified hypotheses to be tested. And I would argue that it's actually inefficient not to have something pre-specified. To, you know, I think a 1,200 patient ad hoc, add-on, you know, the house that whoever built, you know, kind of trial is not necessarily the most efficient trial design. And that an evaluation at a specific time point to determine if your hypothesis is right or it needs to be altered is probably more efficient and more appropriate than, you know, than a sort of open-ended enrollment until somebody decides, hey, I want to take a look at what's going on here kind of thing. And that I think that that would actually aid development. So for instance, response rate was a great endpoint for looking at melanoma with the checkpoint inhibitors, but not particularly good one for lung cancer, right? And so having looked at that early, one could say maybe this isn't the best endpoint. Maybe I have to look at this new phenomenon of a prolonged durability of response and see how best to test this. And it would have maybe truncated the single-arm experience of lung cancer and moved more rapidly into the controlled experience of looking at progression-free survival and those sorts of endpoints earlier. And that's where I think pre-specification of these trials and their endpoints to say, when do I need to make a switch? And when do I need to look at my hypothesis and see why it failed, would be more efficient in terms of a design than what we currently have. I also agree with Steve that we're doing a really, and several of the speakers this morning, that we're not doing a good job of picking the optimal doses. We've had probably about, I don't know, half a decade's worth of experience with continual reassessment models for picking the doses, but I would posit that nobody's really taken a good look at whether or not that's actually yielded better dosing information. But it's probably about time we probably have a critical mass of information that would tell us, are we better at doing that now with this type of model? And I think there are ways that the adaptive designs early will focus us better for the future development and lead to greater efficiencies.
We haven't really talked today at all about drug development for pediatric cancers. And I'm wondering if the panelists think that an accelerated drug development process is appropriate for pediatric cancers, or is there too much concern about risk? Could we envision a multi-expansion cohort trial of any of the drugs that have been talked about today that would include an amendment to add a pediatric cohort based on scientific principles, of course?
I'll be happy to start. And the answer is yes. As we've developed more and more basket studies and looking at expansion cohorts in different groups, the question's been put to us, why isn't pediatric one of those groups in the basket trial? And there really isn't any good reason why it shouldn't be. It will require a buy-in by the existing pediatric cooperative groups around the world. But I think that this is a very nice way of addressing what's been a real concern of theirs for many years, for as long as I've been in oncology, and that is new drugs don't get tested in children early enough. That there is an inordinate delay in testing, therefore any benefit is also delayed. And as we all know, with the vast majority of new drugs that come along, children are actually able to tolerate higher doses corrected for body surface area than adults. So I think that this is something that is very much in our awareness, and we're going to try and make that happen.
Yeah, I would just add, I think that is one of the priorities of the moonshot. Made it to the Blue Ribbon Panel final recommendations. And I think it's kind of ironic if you think about it, because most children go on clinical trials in large cooperative groups, in contrast to those of us in adult oncology who don't do so well. Yet the access to the novel agents is really delayed, as May said. So I think that may change with the new initiatives that are coming forward.
I have a question for Wolfgang. From your experience, you gave a wonderful example of where imaging could identify a flaw in our hypothesis, and that was with iniprib and PARP inhibitors. Did that precede the movement of the drug into phase 3, or did it follow it?
Unfortunately, in this case, it followed it. It was more like a reaction to this failure to think about this PARP inhibitor and how could one image it.
And do you have any examples where you've made an observation that actually has influenced the direction or development or patient selection strategy for an agent currently in clinical development?
For patient selection, I think this whole area of treatment of neuroendocrine tumors with somatostatin receptor ligands and of the whole treatment of prostate cancer with PSMA, radioactive PSMA ligands, this is completely driven by imaging. The imaging was there first, it showed the high accumulation, and then the idea was this could also be used for treatment. I mean, for dose finding, there are a couple of examples that I showed in Dr. Wiesack's slides where it has informed the dose for phase 2.
I have a quick question, just a comment and a question. The comment's for Steve Piantadosi, and I wanted to know if you can design a trial design that will predict the results of elections.
The answer is yes. You just flip a coin.
Okay.
It's almost like a randomized phase 2.
Yeah, I think you'd do as well as anybody else, actually. But no, the question's for Wolfgang. You know, all of us, and many of us see a lot of new patients, and the imaging, your talk was awesome, and you know, that keeps getting more biologic and dual imaging and all kinds of things. But I find even with a plain PET CT scan nowadays, the interpretation that comes with the patient from an outside institution often gets changed when I review with the experts where I work. And I guess I don't know whether or not this is a place where we could emphasize or stress the importance of some uniform criterion if we're going to use these as early markers of response that are going to guide clinical trial design or guide patient care. I find it very frustrating, quite honestly, that the interpretations do change.
Yeah, I really completely agree with this. And in all fairness, probably multiple myeloma is probably the most difficult scan to read on FDG PET. So you may have a negative selection bias here. It's a disease with a lot of variability in FDG uptake and many non-malignant changes that can cause increased FDG uptake. But I think it's very important to have standardized criteria, and that's why I showed the slide with the PERSIS criteria, which is a very simple system, and I'm not saying this is the best way to assess response, but it's a doable way to assess response, and it's published, and one can use it in clinical trials. But you see it very similar to resistance, that it has its flaws, but at least it's standardized, and it should help to get the interpretation of PET scans standardized. I think in lymphoma, it is already fairly standardized how to assess treatment response, although in clinical practice, I agree there's still a learning curve for people working outside larger institutions. But this is really an interpretation of the scans. The scans itself are actually they look the same wherever you get them from. It's just the training of the people reading the scans that may not be ideal for all diseases.
Rich, I have a question for you. You gave the example of the Paloma 1 study changing midway through to incorporate biomarkers CCND1, P16 in an attempt to biomarker select patients who are more likely to benefit from a CDK4/6 inhibitor. And you described the experience that followed. When you look back at that, was that a success or a failure of this approach?
I think if I were to do it again, I might not have done it sequentially. I think that slowed things down a little bit. The way that study was originally designed, it was, all comers, and then once we accrued that cohort, we stopped and we started accruing patients only selected on those biomarkers. But by selecting patients for those biomarkers, we started letting other patients go who were just ER positive, HER2 negative without those biomarkers. And I guess to do it again, and the concern was always if we didn't do it prospectively, we would have had 150 patients, but maybe only 20 of them would have been biomarker positive between two arms. It would have been hard to tease things out. And Maria Kohler's shaking her head. I remember the phone call so well. It's all played out so quickly. But the way I would have done it is probably just take all patients and then either probably stratify for cyclin D1 and P16 and have a little bigger N going forward.
Did you have the tools at the time to do that, though?
Actually, as it turns out, FishPro for 11q13 at least has been available for some time, and similarly P16 is being tested for, I think, in HPV pretty regularly, right?
So this, I think, is a really good example of the question that we face, not having the benefit of 20/20 hindsight, but that we face actually looking forward when we have this decision in front of us. Do we know enough to pull the trigger? Do we know enough to modify the clinical trial design? If we do, are we missing out on a patient population who can benefit? If we don't, are we diluting the effect in the selected group of patients so it will be a false negative study? So this really comes down to the interaction of all the constituencies that are represented in this room about getting as much input as one can in a situation like this. What it means from a practical, from a scientific, from a medical, from an ethical, from a regulatory, from a commercial or payer perspective. All those things come to bear in trying to come up with what you think is the best decision possible given the information that you have and knowing the field is moving rapidly forward. And the longer you wait to make that decision, you're making a decision not to do it. So I think that this is a very good way of ending this discussion, showing that while these principles have really been well laid out by all the speakers and the panelists today and throughout the day, I think that we have to recognize how important it is for there to be communication across our entities so that we reduce the chance of making a mistake as much as possible. Thank you very much.